Okay, so we are nearly halfway through our JH4 syllabus. You'll find that once we come back in term 3, we learn a little bit, then we're done already with your biology life. Some of you may continue on in SH, some of you that will be the last time you take biology. What we covered so far this term we started off with the respiratory system. We also looked at the excretory system. Only 2 chapters, right? Previous term, we looked at nervous system and endocrine. And then before that, homeostasis. Thank you. We learned so far that the endocrine nervous system plays very closely to homeostasis. They are your control systems of the body. Just that one's faster, one's a little bit slower. Other than that, we also learn about the respiratory and excretory system. We learn about how you breathe, how you produce urine. All of this together plays into one big theme, and that theme is the maintenance of life. In your JH3 year, we actually spend a bulk of time just learning about the basics of life. All your biomolecules, enzymes, these are all fundamentals of your life. The things that build up, make the building blocks of who you are as a person. When you came into JH4, we look at how those building blocks come together to form many systems to keep you alive, maintain your living condition. We are now out of this zone. We have learned almost everything we need to know about what keeps you alive, how you breathe, how the blood flows, how you produce and produce urine to get rid of waste. Almost everything you need to know about how your body works. You even know how you are able to see, how you adjust bright light, dim light, how you look at things far away and close to you. Okay? You also learn about how you control the amount of water inside your bloodstream, or the amount of, what else you learn? Or in homeostasis, how you're able to control the temperature in your body. You now know how your body works to quite a deep level. Now we're going to go into your final phase of your biology life as a JH4. This new bulk of chapters relates to this new theme, the continuity of life. We know how life works, now let's look at how we can perpetuate it. How as you as individuals, okay, that's a bit weird, it's like sex ed already. You cannot, and you find a partner. But how do we continue and keep the human race moving forward? We start off a little bit small. We start off with cell division. This is a chapter that is your holiday homework, but we have time, so we will teach and learn together in class. Cell division is reproduction on a cellular level. Individual cells can split into 2, right? And that is what we're going to focus on, reproduction on the individual cell level. When you look at cells divided into 2, that is what we're going to be learning in this chapter. Once we are done with reproduction at a cellular level, how cells divide, we'll then go into the next chapter, the reproductive system. The reproductive system will take place, it will be one of your first chapters in term 3. Once we're done with the reproductive system, no practical, no dissection also. We'll be largely in class. Because I'm very shy, I don't want to teach about body parts, then I'll largely ask you all to go and explore and stuff. No, I'm kidding, I will still go through the parts because I realized over the years that some students don't know what parts they have. I know this because as I teach halfway, then some students during class time, because I'm always observing your emotions, right? Then some students as I teach halfway about reproductive system, then they they cannot believe. There was one year I saw this student look at the anatomy, then don't believe, then whisper into the friend's ears, then the friend's like, yeah, have what? then the student don't believe the friend, then ask another friend, then the friend also say, have what? Then the student was like, huh? Yeah, so some students don't know they have certain parts of the body. Okay, so I realized after that year, I need to educate. Okay, out reproductive system, you will then go into inheritance. Not what you inherit in terms of fortune, property. We're going to look at, the whole point of reproduction is so that we can pass on something. But we pass on DNA and genes. Okay? So that's what the next chapter is about. We're going to learn about how we pass on our genes. How come you look very similar or very different to your relatives, your parents? That is the next chapter. Last but not least, once we know how genes are passed on from generation to generation, then we do one last thing. We're going to learn about genetic engineering. Okay, to end off the chapter, to end off your biology life. Because scientists are at the forefront of biology, we are now able to synthesize, modify the DNA, the genetic code of living things. That is where we are. That is the forefront. We still have many questions because we don't really know how to do it very well. But that is where we will end up the chapter. And then you end up the biology journey. For those of you that want to continue on in SH, what you can expect in SH, I need to let you know. Because some students, after they go SH, they say, Mr. Tan, you love me. I go SH to learn biology, but I don't learn a lot of things I enjoy. If you enjoy a lot of this, unfortunately, that's not what you're going to learn in SH. Okay? Where will you learn all of this? Right? Then it really goes down to what you will study at the tertiary level. People who study this into great depth are students that take biology in university or they take medicine. Then you will learn it to great depth. I have to admit, even as a biology student, I did not study this to great depth at university level. These are for the doctors. Because as everyday Joe, you just need to know how you work. We don't expect you to know how you can fix yourself. Okay? So where will SH topics largely lie in? SH topics are largely here, basics of life, as well as the continuity of life. Okay? So you really want to know if you enjoy these last few chapters, because that is what you may want to make a decision on when you want to take in SH, as well as basics of life. If I'm not wrong, I think in the SH side, they may cover at most like these two chapters. Okay? Okay, so that's where we are, just to paint some context. And today we're going to start off and slowly ease into one of your last three chapters on cell division. When we look at the cell division, if you look at your notes, it looks very daunting, many pages, but you'll find that it's one of the more understandable, easier chapters to do. We will start off, and I think today we'll just spend time at chapter 8, Introduction to the Cell Cycle. If you look at your SLS, you will also be able to follow on with your notes. In your notes, we are at 6.1 and 6.2. That is where the cell cycle, this chapter, part A, is all about. Okay, 6.1 and 6.2. So let us begin. When we look at the topic of cell division, the first question that comes to mind is, why in the world are we studying this chapter? We study this chapter because this is something all your cells undergo, and they are undergoing it quite often. Cell division is a process where the cells divide from 1 into 2. If we can understand how your cells divide, we can understand why some diseases arise, we can understand how to control cell division, and perhaps find some cures in the future. Because when cell division goes wrong, we've learned what happens when everything goes wrong, right? If your nervous systems go wrong, let's say in your eye, you go blind. Respiratory system, if it goes wrong, you can't breathe. Excretory system, if it goes wrong, your kidneys fail, you cannot get rid of waste, you don't pee. What happens when cell division fails? Very often, it's not that it doesn't divide. Very often, it is when the cells divide too much. And that is when you get cancer. So if cell division fails, that's when we get cancer. That's why we want to spend time in your secondary school life to learn about cell division. Because scientists are spending a lot of effort channeled into here to find a cure. Find a way to prevent the cells from dividing uncontrollably, which is what a lot of people with cancer is going through. We just cannot stop it from dividing. Okay? And if it divides too much, we end up getting unnecessary tumors within your body that can disrupt the body parts. So, in this particular diagram, you should find it in your textbook also. It shows you kind of like a life cycle, if you wish to call it, of a cell. Cells go through many phases in their lifespan, and we call it the cell cycle. They go through many things that they do, and one part of their life, some cells will go through a stage where they will divide into two. Not all cells, but quite a lot of your cells will go through this process. And that's what we refer to as the cell cycle. In this first part of the chapter, I expect you to be able to, number one, state the significance of the phases in this cell cycle. Number two, to be able to sequence the phases, to know which one comes after what. Let's watch an introductory video on what the cell cycle is about, what are the different phases, after which we will do a quick quiz.
In this lesson, we'll be looking at the cell cycle. This is the lifespan of a eukaryotic somatic cell. A somatic cell is any cell in the body of an organism, except for sex cells, such as sperm and egg cells. The cell cycle describes the sequence of cell growth and division. A cell spends most of its life in a state called interphase. Interphase has three phases: the G1, S, and G2 phases. Interphase is followed by cell division, which has one phase, the M phase. Together, these four phases make up the entire cell cycle. G1 of interphase is sometimes called growth 1 or gap phase 1. In G1, a cell is busy growing and carrying out whatever function it's supposed to do. Note that some cells, such as muscle and nerve cells, exit the cell cycle after G1 because they do not divide again. A cell enters the S phase after it grows to the point where it's no longer able to function well and needs to divide. The S stands for synthesis, which means to make, because a copy of DNA is being made during this phase. Once DNA replication is complete, the cell enters the shortest and last part of interphase, called G2, also known as growth 2 or gap phase 2. Right now, it's enough to know that further preparations for cell division take place in the G2 phase. Now that interphase is over, the cell is ready for cell division, which happens in the M phase. The M phase has two events. The main one is mitosis, which is division of the cell's nucleus, followed by cytokinesis, a division of the cytoplasm. So at the end of M phase, you have two proper cells, identical to the original cell. The cell cycle describes the life cycle of an individual cell. It has four phases, 3 in interphase and 1 for cell division. Most cell growth and function happen during G1. The cell enters the S phase when it needs to divide. In this phase, the cell replicates its DNA. Replication just means the cell makes a copy of its DNA. In G2, the cell undergoes further preparations for cell division. Finally, we have cell division in the M phase. The M phase consists of mitosis, which is nuclear division, and cytokinesis, or division of the cytoplasm. We'll explore the details of mitosis and cytokinesis separately.
Okay, that was a quick introduction to the cell cycle. To what depth do I need you to understand the cell cycle? Just to that video's depth. Okay, that's all. Don't need to know too in depth of it. We just need to know, number one, whether you can identify the phases. Number two, whether you can sequence it in the correct sequence. Right? And broadly speaking, there are only 4 different parts to the cell cycle. Number one, G1 phase, as you learned earlier on, that's when the cells grow. Okay? Number two, Okay, so this is when the cell grows, right, with its nucleus. So they will grow. Next, in the S phase, S stands for synthesis. This is when new DNA will be produced. Okay, so let's say if you have, let's say this cell only has 2 DNA. In the S phase, what will happen is that it will divide such that each DNA will now have a copy. In the G2 phase, lots of other things will happen. In this phase, new proteins are produced, more cellular structures are produced. Last but not least, the M phase, as you heard earlier on, that's when cells really start to divide. This is where we'll spend a lot of our chapter on, not so much the previous parts. In this part of the chapter, when you call it the M phase, it splits into 2. You heard earlier on, the first part is called mitosis. The second part is called cytokinesis. In other words, if I were to summarize and put it in layman's term, this is when the nucleus divides. This is when the cell divides. It's very interesting because cell divides not in the way we imagine. We think that the cell will divide simply by you cut everything in half. But it doesn't go in that sequence. What a cell actually does is, it will divide its nucleus first. After it divides its nucleus, then it will split into 2. Okay? So that is what we are going to spend a lot of time learning. We're going to learn about how cells carry out the division of the nucleus. And then the very last part, how they split themselves into 2. This process over here, we can lump it together as the M phase. G1, S, and G2, collectively, sometimes you may see people call it interphase. Because it's kind of like the intermission, the in-between where the cell does other things. And then this is where action happens, where they divide into 2. Okay? So take a short pause here. Broadly speaking, this is the overview for this chapter. We will spend a lot of time here. And we will spend a lot of time here. G1, G2, you just know the name can already. And know that G1 comes before S, G2 comes after S. Okay? So let's have a quick check-in. Look at the question below, 1 and 2. Can I get you to quickly try this out? Shouldn't be difficult. Just to warm up for the morning. Yeah, not too difficult. So how many phases are there in the cell cycle? There are 4 phases. Yeah. At which phase does DNA or your chromosomes in the nucleus divide? Divides at the phase that we call synthesis phase. That's where we synthesize new DNA. So as I was saying, we will not spend a lot of time at G1 or G2 because broadly speaking, the gap phases is where, if you can summarize, is where growth takes place. So let's take our attention away from that. Today, we're going to spend a lot of time on just the S phase, the phase where you create new DNA before the cell divides into 2. I have a question for you first. Could you suggest why it is important to divide your DNA to create new copies of DNA before you divide the cell into two. What's the point of it? Is it important? What are the implications if we do not go through synthesis phase and then we just divide the cell and the nucleus into two? I'd like everyone to think about it, then you share your points here, then we can have a look at what you think. Why our body will make our cells go through S phase first. Many points flowing in. You can also read your friends' points on SLS under view all. So why do we need to why do our cells need to carry out synthesis phase to replicate the DNA before it divides its nucleus and itself into two? So some of your sharing from your friends. Minya says the nucleus will have no DNA inside if we do not carry out this process and divide. Zuni says if not, the total amount of cell will just keep decreasing. You mean total amount of DNA in the cell? Yeah, will just keep decreasing with every division. Makes sense? The replicated cell might be missing parts. What do you mean by parts? How young?
Like the DNA.
Okay, so if we do not replicate our DNA, as we divide, we maybe end up missing some DNA as we continue division. So that each cell has enough DNA after the division. I like the choice of word enough. It implies that we need a certain amount of DNA for each cell to function. That's true. Each cell is only able to function if it has its full set of DNA. Because remember what's in our DNA? We learned in JH403, transcription, translation. DNA is a library, a genetic code library that tells our cells how to produce every protein in the body. If we are missing some, then we won't be able to carry out and synthesize all the proteins we want. So that the cell has enough time to prepare for cell division, instead of cramming and rushing everything in M phase. This is interesting. So for Sawyer, she thinks that the synthesis phase kind of gives you a buffer time before you do everything here. Oh, sorry, are you saying that instead, if we try to do this in here, it's very rushed? Okay, I've never quite thought of it that way before, but I have to agree. If we try to do this as we are dividing the nucleus and as we are dividing the cell up, it's actually not enough time. This phase over here takes place very, very, very fast. Okay, but this part over here, we must do it slow and steady, very carefully, because if we don't divide our DNA properly into two, you can end up with mutated DNA. Shermaine says if synthesis doesn't take place, there won't be enough genes in each daughter cell after the parent cell divides. I think Shermaine has read up a bit more, so she's using some terms like daughter cell, parent cell. The daughter cells are simply the cells that you get after one cell divides into two. Each one of these are called daughter cells. Okay? And then the original one is the parent cell. Okay, let's see your other friends' responses. Each one says the DNA will be split into two different things, and the new cells might not contain the same genetic information. Fair enough. If we don't divide into two, then how are we going to share the DNA between the two cells, right? Instead of the remaining cells, one cell will take the eyeball genes, then the other one take the nose genes. Yeah? You can imagine if you would produce sperms and eggs, each one will take a little bit of each parent. eyeball, nose, if it fuse back together, I don't think you'll get a full human being. Since he says easier for the cell to divide with a nucleus ready for a new cell. That's true. I have to agree with you. Actually, all of this here is prep work. We prepare so that the nucleus is good and ready for just to divide into two. Kyra, the daughter cells will be identical, which could threaten the species' chances of survival. Kyra, could you elaborate a little bit more about how synthesis will affect whether it's identical or not? Okay, I understand the importance of having genetic differences. I think, okay, then my question to you is, are you implying that under the synthesis phase we could generate new DNA? Different versions of it. Okay, I think there's some truth in this. During synthesis phase, yes.
What if there's something wrong in the S phase and the DNA doesn't replicate properly, then won't we create like faulty cells?
Okay, so we combine Finya's point and Kara's point together. Can new versions of DNA be created in the S phase? Okay? The goal of S phase is to produce a new copy of the DNA that's exactly the same. Okay? Exactly the same. If not, which is what cancerous cells are like, if not, in your normal body, what will happen is, let's say you get a cut somewhere. Your skin cells over here will divide to produce new skin cells that are still skin cells. Yeah? That's because we want everything to be identical. The moment it starts to be not identical, things go crazy. So then the question is, during synthesis phase, can new versions of DNA be created? And I like to combine it with Nia's point. Can. New versions of DNA can be created as we are creating new DNA if mistakes are made. So Finya asks, what happens if mistakes are made? Oh, okay. If mistakes are made, actually we will create new versions of DNA. New versions of genes. It can be good or bad. Sometimes it's good, sometimes it's bad, sometimes it has no effect. For example, our eye colors, actually they are all the result of mutations happening during the S phase. But it doesn't really impact us, just change color only. But can other mutations take place here? Can. As we are trying to make new DNA, the new mutation could result in, let's say, one of your organs not functioning that well. Then, the person may not survive long. Yes, yeah?
Okay, then is that the Meselson and nonsense mutation?
Oh, yeah, so last year you all learned all the missense, nonsense, all those kind of stuff. Where do these mistakes tend to take place? It tends to take place during the S phase. Thank you for bringing all of this in. Okay, so last year you learned missense. Forgot to spell missense already. Nonsense. What else you learn? All your frame shift. Remember all these mutations? Yeah, they tend to happen during the synthesis phase. As the cell is trying to diligently copy and make new DNA, sometimes they make mistakes. Sometimes sometimes your copy notes on the whiteboard also write down different things, right? Same for ourselves. If they copy down different alphabets, they replicate differently, then that's when all the mutations can occur. And they tend to happen during this phase. Can be good or bad. But in instances where it does not have any bad effect, we see it among all of ourselves. For example, we all have different heights, different skin color, different weight, different eye color, different hair color. All of these mistakes, sorry, I'm making us sound like we are mistakes. All of these different versions of each other, all the characteristics, originate here when all of this happens. Can bad mistakes happen? Can. Often, these individuals won't survive to tell the tale or to pass on the mistakes to the next generation. Okay? But the fact that we are here is because they're not harmful. We pass on whatever we have or what you see on the surface. Jeanette asks, by replicating the DNA, there will be enough DNA to be split. Agree with you? If we don't replicate, then how are we going to split the assets, the fortune, right? Kiran, to ensure that genes are passed down to the next cell. Agree? We don't split. Don't know how we're going to split and share the genes among each cell. Ella, because S phase is responsible for DNA genetic material of the cell, so we need to replicate DNA first so that each cell will have the same genetic material before the nucleus splits. I think this is quite a good summary of a lot of the points our friends have shared. Teacher, the cell needs to have enough DNA to divide into two, so that's why we need to replicate the same amount of DNA. Yeah, it is true. Shreya, the replicated cells may not have enough DNA inside. Okay, so I notice our explanation goes both ways. We look at it from the perspective of if we do not divide, what will happen, or why we need to divide. And so our friends they say, what happens if we do not need to? If we do not divide, this is the outcome of. If we do not divide and create new DNA, each cell will have less and less and less DNA. So it says to allow new cell to function, and we learn that the DNA is inherently tied to many functions in your body. Nabila says so that the DNA can be passed down to the other nucleus, which is in the next daughter cell. Alpha, since DNA inside the nucleus, it's better to synthesize DNA first before the nucleus splits. I think this aligns with what a lot of your friends are saying, that the DNA is inside the nucleus, so it makes sense to divide the library first before we split up the whole nucleus into two. If S is after M phase, the DNA might need to find its way. Oh, that's very interesting. Okay, that's a very interesting train of thought. If we park this after the M phase, then if we divide the nucleus already, then divide the DNA, then somehow the DNA needs to travel to the next nucleus. Okay, very interesting. Once it says the S phase allows enough DNA to be split into daughter cells. So, as you can see from all of your suggestions, indeed, we need to carry out S phase first so that we have enough assets, fortune, enough information to pass on equally to both cells and of the exact same copy. Because we want each cell to be able to do exactly the same thing as the parent cell. Your friends have also suggested that if we don't do so, we will end up with cells that have less and less and less DNA and become more and more and more non-functional. People with tumors, the cancerous cells, if you look into each one of those cells, some have more DNA, some have less DNA, because the division is out of whack already. They are not able to even control how they divide. That's why they divide uncontrollably. Unfortunately, there's something that we have not found a cure to. As people age, become older and older, cancers become more and more rampant. For lucky individuals, they may grow old and never have to encounter cancer. But actually, if you look at the statistics, older people, the older you go, the higher and more risk you have of getting cancer. Why? Because actually, as you grow older and older, your cell's ability to regulate their own life cycle becomes very bad. They become very bad at even doing simple things like replicating the DNA into copying it down word for word. They cannot do that anymore. And once we don't copy word for word, things go out of control. So that's why we tend to find cancers appear in older people more. So that there will be enough DNA for the next cell. Have I talked about Kai's one? Yeah. So Kai sums up the whole thing. So that's where we are. We know the importance of the S phase. The next part of this chapter, we're going to look at how our cell carries out the S phase. To be able to remember to learn how our cell carries out the S phase, it is important that we first do a quick recap on the structure of DNA. Do you still remember the structure of DNA? It links to your JH403 topic. How much can you remember from the structure of DNA? If you give me some summarized bullet points. Give you some time now to recall. I've only drawn 2 lines on the whiteboard, but I think we can gather a lot of things from the structure of DNA. How do you recall the structure of DNA?
So DNA is ATG.
Okay, so we started the ball rolling. Double helix, that's why you said. Okay, first characteristic, it is a double helix. Actually, we have 2 pieces of information here. Helix represents the helical structure I've drawn here. That's what the helix means. Double means that there are 2 strands. So I need you to bear this in mind as we go through this chapter. From now onwards, when I say a DNA molecule, what I am saying is a DNA molecule with two DNA strands. I say again, from now onwards, if I use the word DNA molecule, remember that each DNA molecule has two DNA strands. The word molecule is not equal to strand. Different things, okay? I say again, if I say a DNA molecule, I'm referring to the entire thing. Both strands included. So, in other words, another way I can say is, how many DNA strands are there in a DNA molecule? There are two DNA strands in a DNA molecule. Okay? And so we gathered quite a lot from the word double helix. Anything else you recall from last year? It is made up of nucleic acids. That is where the name comes from. Deoxyribonucleic acids. So what is the base unit of DNA? What is the monomer? What is DNA made up of? What's the individual unit? Starts with N. Yes, there's nitrogen inside.
Nitrogenous bases
Okay, Ken. Nitrogenous. So Finya built on alpha point nitrogenous bases. Okay, there's nitrogen inside. Maybe another word that you recall is nucleotides. That's what the individual units of DNA are. That's what their repeating units are. Nucleotides. More specifically, DNA has a special kind of nucleotide called deoxyribonucleotides. Many of them put together, you end up getting DNA. A deoxyribonucleotide. Any other properties of DNA you remember? Anti-parallel. Okay, I draw your attention to what I'm about to write on the whiteboard. One strand goes in the 3' to the 5' direction, the other one goes in the 3' to 5' direction. They are anti-parallel. Okay? I will not spend a lot of time recapping why one strand, what constitutes 5' and what constitutes 3'. Okay? In summary, you know that as DNA nucleotides join together, there will always be some free ends, right? The free end is what defines whether it's going to be 3' or 5'. Okay, but we don't have to focus too much. Any other properties of DNA? I think there is still one more. I'm not filling the blanks in between the DNA. Yeah, anti-parallel. The 5 to 3, 3 to 5. This anti-parallel. Notice my DNA is rather bare in between.
Hydrogen bond.
Okay, there are hydrogen bonds, but between what?
Between the A and
All the ATCG, remember? Okay, so bear in mind, you also learn about AT, you also learn about CG, and that's how they hydrogen bond to each other. Yeah? What Finya was talking about, these lines I'm drawing, these are all the hydrogen bonds. And then you have all your ATCG in between the DNA. What we refer this to is complementary base pairing. These are all recapped from my JH3 years. Okay, complementary base pairing between AT and CG. Broadly speaking, these are the properties of DNA. They are just like to recap from your JH3 time. Okay, it provides you with enough foundation so that we can understand how our DNA replicates. I'll pause here for a short while. Yeah? Okay, I'm going to erase the whiteboard. Anyone wants to take a photo or write anything? Why? Yeah. I mean, this is just recap for your JH3 years. Okay, nothing new. Okay, so I'm going to erase the whiteboard. Give me some time. I'm going to set up and draw some stuff on the whiteboard first before we dive into the final part of today. I think we won't do too much since we still have time tomorrow. Okay, give me some time, I'm going to set up the whiteboard. In the meantime, you could read parts of the chapter to learn a little bit more, or you can just take a mental break. Okay, I am ready. Right now on the whiteboard, what I'm going to be going through is a very important experiment in history that helped scientists figure out how our DNA replicates. We know so far that our DNA, a single DNA molecule, is double-stranded. Yeah? A single DNA molecule is double-stranded. But actually, scientists back then didn't quite know how our DNA replicates. Granted, any process in our body needs enzymes to carry out the process. DNA replication at its core happens because there are enzymes that carry out the process. Just like anything that takes place in your body. Your seniors used to learn exactly how the enzymes will replicate the DNA. We've removed that out of your syllabus because we realized it's really just useless info that you're memorizing. Okay? So instead, we decided we'll spend a little bit more time looking at the history of how scientists figured out which model of DNA replication really takes place. When I refer to models of DNA replication, I'm going to show you what I mean by different models. Long time ago, there were 3 different models of DNA replication that scientists were arguing between. I'm going to focus only on 2 of the models in history. The 2 models go something like that. Now we are going to take out my props inside my. Okay, the 2 models go something like that. The first model of DNA replication that scientists was hypothesizing is called conservative replication. And you see what I mean and why they call it conservative replication. We know that DNA is made up of 2. A DNA molecule is made up of 2 DNA strands, right? So I'm going to use my magnets to represent that. I've written the word original so that we can always trace where the original parent DNA is. So this is a DNA molecule with 2 strands. This is what scientists speculate. They used to think that DNA molecule, in order for them to be replicated, number 1, they think that number 1, it needs to be split up. Each one of these DNA molecules. Okay, so what I'm trying to do here, I'm trying to show across time. So this is the process that's happening. They think that after it splits up, the new DNA that is synthesized will be synthesized like that. Okay. Each original DNA strand serves as what we call a template. So this is a template. So that's what scientists think lah. That the original, each strand will serve as a template to synthesize a new strand. And enzymes are likely involved in the process. But what they think happens, and that's why you see why it's called conservative replication. They think that after the new strands have been created, the new strands will detach itself from the original strands and bind together, and the original strand will come back together. That's why we call it conservative replication, because scientists think that in this model of DNA replication, the original DNA molecule will be intact after the whole process. After it's split up, create new one, it will join back together. While the new two strands will join together to form a completely new DNA molecule. That's why they say they call this model conservative, because it conserves the original DNA molecule. This name makes more sense if I show you the contrasting model that they had in that time. They called the other model semi-conservative replication. Because another group of scientists felt that, they think that it's not so efficient to do this. They think back then that each one of these strands, they agree with the front part, that each one of these strands will be used as a template. Each one will be used as a template to synthesize a new DNA strand. But where this group of scientists differs here. This group of scientists felt that the DNA molecule will not detach and rejoin to form one completely new and one completely original. Instead, this group of scientists felt it made more efficient sense if the new and the old DNA strand joins together to form a DNA molecule that is half original and half completely new. That's why we call it semi-conservative, because only one of the strands is old, the other strand is new. Okay? So these were the two conflicting models of the time. And scientists didn't know how to find out the truth. Because all of these things are microscopic, worst part is you cannot even view these processes taking place because it takes place in your nucleus. How do you break open a nucleus, scan and look at your microscope inside to see this happening? Very hard. But two scientists called Meselson and Stahl, which the experiment is named after, in biology is also known as one of the most beautiful experiments ever done. They call it the Meselson and Stahl experiment. This experiment is very, very, very smart because these two scientists found a way to figure out which model actually takes place. To understand which model actually takes place, we need to learn about we need to learn about the very ingenious idea they had. Earlier on, Finya and Alpha alluded to the idea that our DNA is made up of nitrogen, right? Other than carbon, oxygen, hydrogen, there's nitrogen. One of the defining things of our DNA. DNA has nitrogen. That is the key thing that these researchers decided to use. Because they realized, and I'm sure you learned in chemistry you learn that every element has isotopes, right? Every element has isotopes, and nitrogen has isotopes. Two versions of it, nitrogen 15 and 14. You all learn in chemistry, if you just look at this, nitrogen 15 and 14, you know which one is a heavier nitrogen. Makes sense, right? Because the 15 there is because it has extra proton or neutron. Should be neutron, right? Because the number of protons defines the element. So it should have more protons. Nitrogen 15 has one more neutron, which makes it heavier. Yeah? With this piece of information, the scientists managed to come up with a very interesting experiment. The experiment goes something like that. They worked with bacteria called E. coli. Why they decided to work with E. coli? Because E. coli divides very fast as a cell. Unlike human beings, we are an entire multicellular organism. This is unicellular. So it divides extremely fast. Just give it a few hours and it will divide fully already. So the interesting experiment they started off doing was this. They had a petri dish. Inside their petri dish, they had E. coli, and this is what they did next. They then went on to put the E. coli and they mixed it with nitrogen 15. And as the E. coli continued to grow and develop, and the population increased, what they found was that all of the E. coli ended up having DNA that was made up of nitrogen 15. Okay? So all of these bacteria here has nitrogen 15 inside their DNA. The DNA is very heavy. Okay? So they just tested a concept. It is possible to make DNA change and take up different isotopes. So imagine now, if I perform the same experiment with E. coli, a few of them, and instead I go and put in and let them grow in nitrogen 14, what you'll find is that as the E. coli population grows, more and more of them will have nitrogen 14 inside their DNA. A lot of them, their DNA will be very light. Yeah? So that's what they just tested. They tested and realized that we can make use of nitrogen 15 and 14 to change the property of the DNA. Make them either very light or very heavy. But that's useful information because that's what they use to find out which model actually took place. Okay. The next thing they did was they used a special machine called a centrifuge. A centrifuge is a device that uses kind of a test tube. And what they do to the test tube is they will collect whatever samples they have and then they'll spin it. So let me show you what a centrifuge kind of looks like and how it works. A centrifuge machine works something like that. Maybe you learned this in chemistry before. If we were to put liquid into a centrifuge, and then we put it and let it to spin, after a while, you will find that the layers will separate. Heavier things will go to the bottom, lighter things will go on the top, the harder you spin. They use this same technique on the bacteria. They take the bacterial population out, they kill it and smoosh it, put it in water, then they take it for a spin. And the interesting thing is, after they took it for a spin, they found that because the DNA here is heavier, it ends up sinking somewhere to the bottom. Makes sense, right? And the DNA here, because it has been growing in nitrogen 14 for many populations, when they took these bacteria after killing them for a spin, the DNA kind of ended up at the top. Make sense so far? But with just these two pieces of information, they figure out a very smart way to carry out an experiment to find out which model is the correct model. Because now they realize, actually I can tell the properties of the DNA, whether they are heavy or light, just by doing this centrifuging. Okay? So this is what they did. They decided, okay, let's work this out. They decided now to carry out and grow bacteria in these two different kinds of medium. So this is what I'm going to call generation 1, generation 2, generation 3. Imagine now, let's grow all of our bacteria in nitrogen 15. Okay? We grow all of them in nitrogen 15. Agree with me? After a long period of time, you'll find that all of the bacteria will have heavy DNA. Okay? So all of them will have heavy DNA. Yeah? If I were to kill all of these poor bacteria over here, take them for a spin, what would you expect inside the test tube? All below, right? Okay, they're all below. Nothing quite spectacular. This is what we expected so far. Okay, it's the next step that they were very smart. Because they realized what if in the next generation I do something different? What if I take some of this bacteria, put it in a new petri dish, but this time I grow all of these new bacteria inside nitrogen 14. Just by changing this one step, they managed to solve this mystery. It's very smart because you see why? If this model is true, it means to say that you will have DNA that is heavy, and you also have DNA that is light. What would you expect to see in the test tube? Agree? You will end up finding one band at the bottom, one band on the top. If this particular model is the correct model, right? So this all in theory, they haven't conducted yet. But in theory, they speculate. If this model is true, we should see one band at the bottom, one band at the top. Very smart, right? Just by changing which medium it grows in. Let's look at this particular model. Same thing, let's start off first with growing all our bacteria in nitrogen 15. And what you expect is, if you go and take this for a spin, it will be on the bottom, correct? But this is where the difference lies. Okay, so we agree at the start they should all be like that. Okay, but if it's semi-conservative model and we take some of these bacteria, go and put them instead in nitrogen 14, something different will happen. We will find that the DNA will be something like that. Because in a semi-conservative model, one strand will be new, one strand will be old, combined into a single DNA molecule. This is what all the DNA molecule will look like. If we take all of these DNA molecules and go for a spin, where do you think the DNA band will lie now? I have a few locations. Do you think it will lie at the bottom and top together? Okay, I heard teacher say, you think where you like? Middle. Teacher is right. In theory, if I take this for a spin, if the DNA does not split up into two and stays together, in theory, all of them will end up somewhere in the middle. Because now each DNA molecule is half light, half heavy. So it neither sink to the bottom, neither go to the top, it will be somewhere in the middle. So very sharp observation by Eaton. But that just means Meselson and Stahl, if they conduct this experiment, they just need to see in the second generation what the pattern will be. Right? If they notice the pattern is split like that, then it's conservative. If the pattern is right in the middle for the second gen, it's going to be semi-conservative. So which pattern did they see when they carry out this experiment? They actually saw this particular pattern appear. And they were very happy because their theory worked out. When they carried out the experiment, first generation is what they expected. Second generation, when they saw this band, they were very excited. Because then they realized, they found out that in actual fact, our DNA replicates in a semi-conservative way. Not in this inefficient way, but after replicate, it will split up again and rejoin again. Yeah? I was quite pleased to know that I thought this experiment was done so long ago until these people don't exist. I think some of them are still alive. So when I went to YouTube, I actually managed to find a very, very cute and nice video of Meselson and Stahl themselves talking about their own experiment. They are two cute, very old men now. Okay, you scroll right down to the website under Stay Curious. Let's see where they have up their video. Yeah, video away. Oh, okay. So this video over here, let's say you don't like the way I taught it, and you want to hear from Meselson himself about how he decided to do this experiment, how he conducted it. I found this lecture online conducted by himself, Meselson, this old man over here. It's quite special to be able to watch someone who discovered this process and they came up with this experiment explaining it themselves because they are very passionate about their own project. When I explain how I'm only like maybe 1% of what he is. This is Meselson explaining his own experiment. I have another cute video I can't find a video. Oh, over here. The most beautiful experiment, Meselson and Stahl. These are both of them.
John Cairn sat on the telephone and very excited was he had just read Mendel's papers. And you know, they are the most beautiful
Meselson?
experiments in biology.
Stahl.
And I guess. This is John. John,
It's cute.
you said.
I have not Googled recently. I don't know if they are still around or they pass on.
Did I?
But this was around 3 years back when I Googled them. So this is that. You have time, you can, because it's a very long video.
They proposed a model. There are those who believe this model must be true because it was so good. And there are those who believe it must be wrong because biology is complicated and this model is too simple to be right. Did you see? Yes. But there was no experimental proof of it. They had a model which made a distinct prediction about how DNA replicates. And it needed to be tested, and it's fun to test the hypothesis. We agreed that we would go to work together to figure out whether or not it was right. When Frank and I showed semi-conservative replication, it wasn't just a model, it was something real like that. After our experiment, it was now widely accepted that their model, Watson Crick model, is right. So it became the building block, you might say, for all of biology. Yeah. From very early childhood, I mean practically infancy, I love science. I love to put wires together to make little radios, which I could put under my pillow so my parents wouldn't know that I was listening to them all night long. And then I was very interested to know what makes life work. And there had been, I think, in the house, maybe in my bedroom, that painting by Michelangelo, the God and I, Adam, well, and the touching figures. And I don't know if there is a spark. I don't think there's a spark in it, but I'm not sure. But to me, that life is somehow electric. That God is providing life through a spark. And for some reason, that made me interested in electrochemistry. Unlike that, I had no particularly strong interest in science. As it was understood that when I graduated from high school, I would apply to the Naval Academy. That's what my mother had in mind because she thought I would look good in dress flights. But then, of course, World War II broke out, so that plan changed fast, and she decided I should just go to college. I think I was just too young to understand the courses in humanities. I hadn't had enough life experience to get a grip on the questions they were even thinking about. Science, on the other hand, was concrete. Children can grasp science. And among the sciences, biology was the most appealing. There the one was that you could figure out puzzles. That is, there was a rational, concrete, quantitative explanation for what you saw. You could reason backwards as to what must be going on. And that intrigued me enough to know that genetics was something perhaps I could do.
So I will pause here. You can see that your life journey shaped by many, many steps in life. Don't need you all to be very interested in biology, but just take your interest, pass it on to someone else, because they may be the next scientist that may do something quite amazing. Yeah? So, you can watch this whole documentary if you like to, because I think it's very cute and very interesting to hear it from the experimenter's point of view. To end off this part of the lesson, if you understand this model, let's take it to a third generation. Generation 3. can you predict what the bands inside will look like? If you think you understand the process so far, can you predict what the bands will look like? If I place the bacteria and let them grow again in another medium still containing nitrogen 14, what do you think the bands will look like? Okay? If you think you understand, try now. How many bands will you see? Where will the bands be?
Wait, 4 pairs, right?
4 bands ah?
No, 4 pair. Eh?
How will it be different if it's conservative and semi-conservative? Will some bands be thicker than the other? Everyone gave it a good mental exercise? Okay, let's look at conservative replication. The idea of conservative replication is that whatever was present, okay, so now I'm splitting it up. So these are my original strands from Gen 2. Okay, these are my original strands, all will use a template, right? Okay. We will then, since they are growing in N14, any new DNA that's synthesized will be blue. They will be light, correct? Following? Okay, but then because it's conservative, we learn that the new strands that are synthesized, they will rejoin back together, and the old strands will rejoin together. Similarly on top, the new strands will join together, while the old strands will rejoin together. Hiya.
But like, it's two things. Like, two things is like one, like it makes a new one. Then where does it make it from?
Oh, where does it make it from? Okay, so it makes it from nucleotides. Okay, so when I say I'm adding N14 or N15, I'm adding in, now I'll be more specific, I'm adding in nucleotides that contain nitrogen 14 or nitrogen. Better? Yeah? I agree with you, I just toss in nitrogen, how is it possible to make DNA? Right? So I'm tossing in nucleotides that contain N14 or N15. Okay, so this is what you end up with, right? What would the band look like? You will have a heavy band. Then actually you should have a lot of light DNA. Yeah? Okay, let's look at the semi-conservative one. We know in semi-conservative, each one of the original strands, same thing, will serve as a template to synthesize new DNA. New DNA, new DNA, new DNA, new DNA. Except that in semi-conservative, don't need to separate and rejoin. Just leave it as it is. So what would the bands look like? You'll find a band on top, and you should find a band in the middle. And they should be of the same thickness, cause equal amounts. Yeah? If we go to a third generation. Yes, Haya?
But like, when in the test, will they ask for conservative?
Ah, okay. So, since we already know for a fact that this is what is true, right? Is it possible we still ask you a conservative model? So, that's why for this particular experiment, don't memorize the experiment, understand the experiment, because what we tend to do next is that we will use the concept of this experiment, but we will change many factors. This is how we could change it. We don't even say conservative, or we will usually tell you which model. We will say use conservative or semi-conservative. Other than that, we will also maybe change things up. We may say, maybe second generation we put N15. Then the third one, then we put N14. Maybe in the third one, then we put N15. How will the bands change? So that's how we will test your understanding, by changing factors. But we will tell you whether it's conservative or semi-conservative, because if you don't know, then you won't be able to decipher what the bands will look like. Teacher, you got a question? Oh, you understand now? You want me to run through?
What about Gen 4?
Yeah. Gen 4, I mean, you just continue on. Okay, so Gen 3,
Where's
teacher, so this is Gen 3. Gen 2 is like that, right? 2 DNA molecules, right? Imagine if I split up the DNA molecules, split them up, now I have 4 templates, right? Okay, so because I'm growing it at N14 DNA, so now every new strain that's synthesized will be light. Yeah? So now I have 4 DNA molecules. Some of them are fully light, some of them are half light, half free. So that's how I get my bending patterns. So Pinya asks, what about Gen 4? So you see, I can go endless number of generations. If you understand the concept, then you should be able to figure out 4, 5 onwards. For those of you that don't understand what I've just ran through, you have 2 alternatives. Number 1, on my website, I've recorded myself teaching the experiment from start to end.
Hi everyone, in this
Okay.
video I will be reviewing
So let's say you really don't understand what I said, I taught the same thing here.
two specimens
Okay. So that's where first location you can find this. Second location you can find it is inside your SLS. Over here. Okay, wrong. Over here. Okay, that's where you can find it also. Okay, for your recap and your learning. This experiment, some students find it very challenging. But I hope that as you saw from the whiteboard, that it is not challenging, more elegant than it is challenging. Just a very simple concept used to solve a very big mystery that one cannot even see on a microscopic level. Okay? Okay, I'll pause here. I think that is enough for today. Okay, you can take some time if you like to. There are some questions that you are able to try already. Under practice makes progress, there are 2 questions. Question 1 is an MCQ. If you like to, you can try this MCQ already to get a flavor of what kind of questions you can ask related to this experiment. You'll find that it is we play with the experiment. Then you see if you can understand based on the concept we've learned. Okay, so that's all I have for today. You can take a pause already. Y'all can go for a short. Can y'all go for like a 10-minute break? Let me figure out what is supposed to happen in the time.